Hello, my name is Jacob Stewart from Connecticut College, here with my talk for the 2021 virtual ISMS. I'm presenting this work also on behalf of my undergraduate students, Jacob Allan, Melanie Fournier, and Grace Kovic. And we're going to be talking about sensitive infrared spectroscopy of isoprene at the part per billion level. Isoprene is an important molecule in the atmosphere. It is one of a class of molecules known as biogenic volatile organic compounds. These are present at low concentrations in the atmosphere, but are an important part of atmospheric chemistry. Isoprene in particular, C5H8, this molecule here, is about 50% of all of the biogenic volatile organic compound emissions annually. There are other molecules also emitted, such as the other ones listed here, alpha and beta pinene, limonene, and many, many others. But it's important to understand the properties and concentrations of isoprene in particular, as well as these other molecules. And one reason for this is that isoprene, with its two double bonds, is a very reactive molecule. And so isoprene in the atmosphere undergoes oxidation with molecules such as ozone and nitrogen oxides. And in the process, it turns into these oxygenated processed BVOCs. So we have here methyl vinyl ketone and methacrylene as examples of that. And through the process of this oxidation, the volatility of isoprene is decreased. And this leads to two things that we care about in the atmosphere. One is the formation of aerosols. So the lower volatility means that these molecules will start to condense and form solid particles. And it has been shown that isoprene and other BVOCs, upon oxidation, will turn into aerosols. And that's important. Aerosols play a really important role in the atmosphere. And also, in the process of oxidation, if there are nitrogen oxides in particular present, isoprene can form tropospheric ozone. Not directly, but as sort of as a byproduct of this oxidation process. And the presence of ozone in the troposphere is bad. It's a component of smog. It's a pollutant. We like it in the stratosphere, but not in the troposphere. So that's those are a couple reasons why we're interested in studying isoprene in the atmosphere. Isoprene also shows up in our breath. Isoprene is one of many different volatile compounds found in breath. And there's interest in measuring breath isoprene as a way, a non-invasive monitoring of patients. And this is because isoprene has been linked to the cholesterol biosynthesis pathway. And also, in particular, it's been shown to have promise as a marker for advanced fibrosis in people with chronic liver disease. So these are some of the reasons why why it's important to be able to measure concentrations of isoprene at very low levels, because in both the atmosphere and in breath, isoprene is present in the part per billion range. And so we, in our lab at Connecticut College, are working on developing an infrared spectrometer that can measure these low concentrations of isoprene. And that's the work we're presenting here. There are other ways to measure isoprene, for example, mass spectrometry, and this is a complementary technique to those other methods that already exist. And so the goal of our work is to use infrared spectroscopy to measure low concentrations of isoprene. And the basis of our work is essentially, any other absorbance spectroscopy, the Beer-Lambert law, that the absorbance is directly proportional to concentration. Now, because the concentration is so low, we need to, this value of C is small. We need to get to be able to observe absorbance. We need to have as large a value of epsilon and beta as possible. So epsilon tells us how strong the peak is, right? So the concentration is very small. And so we want to use the biggest peak we can. And so in the region of the laser that we have to use, around 990 wave numbers, this is the peak right here. So this is the Q branch of the new 26 band of isoprene, just above 990 wave numbers. And so that's the target peak we're using. And the other nice part about this is it's well away from absorption of water and CO2, though you will see CO2 does show up, but the peaks are very weak. There has to be a lot of CO2 for it to be present. The other thing we can do is increase the path length in our measurement, and we do this using a multipass cell. And so this is a picture of the cell that we have in our lab. So how do we do these measurements? This is our instrumental setup. We use direct absorption spectroscopy to measure isoprene, and you do it in a multi-pass cell. So we use a quantum cascade laser. This is a Daylight Solutions external cavity quantum cascade laser. It's driven by a piezo driver and function generator driving that to modulate the frequency of the laser. Light from the QCL is sent through a reference gas cell that contains methanol for absolute frequency calibration. And then for relative frequency calibration, we use a germanium etalon. And then we have detectors to see the light from both of those. The rest of the light is sent into our multipass cell. The picture of it is down here, which has a 76 meter path length. And then the light that exits the multipass cell is sent onto another detector. And so this is how we do our measurements. So our first thing that we measured was a certified sample of isoprene. So air gas, we called them up, and they were able to make a mixture of isoprene and nitrogen for for testing purposes for us that contained 10.8 parts per million by volume, with an error of plus or minus 0.5 parts per million. And so we put this sample into our spectrometer, measured it, and compared it to some previous results from Brower et al., from a group at the Pacific Northwest National Lab that had measured the absorbance values of isoprene that had established absorbance cross sections of isoprene across the infrared for purposes just like this. And so here's our data. We have in red, the red trace here. This is our measured data, and it's at much higher resolution than the data from Brower et al. The black circles here are the measurements for Brower et al. Scaled to the concentration that we have, this 10.8 part per million by volume at atmospheric pressure and room temperature, and then also scaled for the path length that we have for that 76 meter path length through our multipass cell. And so we can see really good agreement between our measured values and the predicted values we would get from using the data from Brower et al. And so that gives us confidence that we know the proper path length through the cell, and that we have a good match between previous results and our results. So the next thing to do is that, of course, the samples that we eventually want to measure are much lower in concentration than this 10 part per million sample. And so we want to measure lower concentrations and make sure that we see that linear behavior that we'd expect based on the Beer-Lambert law. And so to get to lower concentrations, we mixed our certified isoprene with nitrogen to dilute it. And we used valves, needle valves, to control the flow and measured the flow using a couple of flow meters pictured here. And then that got sent into the spectrometer, flowed through the multipass cell, and then exited into a vacuum pump. And so with that, we were able to control the concentration and know the concentration of isoprene as we diluted it with nitrogen. And so using that, we were able to obtain measurements where we retrieved concentrations. We have our measured concentrations on the y-axis here. This is from least squares fitting of our data, compared to our known result. We can basically scale that based on the Beer-Lambert law and retrieve concentrations. And then we have our nominal concentration here, which is what we get from using the flow meters to determine how much the dilution factor is. And so we see very good agreement between what the concentration is based on our flow meter measurements compared to what we retrieve from our spectrum. And so we see a very linear behavior, a slope of essentially 1, and y-intercept essentially 0, which is what we would like to see, what we would expect. So this is showing good results for our spectrometer. The next thing we'd like to measure, that 0.34 parts per million that we went down to, that was the lowest concentration we were able to make. That was limited by the measurements we can make with our flow meters and the control we can have with our needle valves. And so what we really want to know is what's the lowest concentration we can measure? What's our detection limit? And to do that, we measure the Allan deviation. This essentially will give us the noise equivalent concentration. And so we measured the same sample over a period of over 1,500 seconds, almost 2,000 seconds. Each of these crosses here represents an individual measurement where we retrieve the concentration using least squares fitting. And so we can see there's a bit of drift in our measurements, and that shows up in the Allan deviation as well. But what we're interested in here is what is the lowest value on this Allan deviation plot. So on the y-axis here is our Allan deviation in parts per billion. So this is the effective concentration that's equivalent to the noise in this series of measurements, which tells us what the detection limit essentially is of our instrument. And on the x-axis here, we have this based on measurement time. And so if we average for about 10 seconds, we get down to a limit of 3.2 parts per billion. So that is sensitive enough to do breath measurements, but not sensitive enough for atmospheric measurements. Those atmospheric isoprene is on the order of 1 to 10 parts per billion, which would be right at the limit of, the higher concentrations would be right at the limit of what we could detect reliably. Lower concentrations are below our detection limit. Breath isoprene is a little bit higher, and we'll see we can actually observe breath isoprene. And again, as I mentioned, there's a large amount of drift in our instrument, and that shows up in this Allan deviation swinging back up. We believe this is probably caused by mechanical instability, based on, and we tried to get rid of this as best we could, but we're unsuccessful in doing so. One other issue we're going to run into is, so all the measurements that I've talked about up to this point, we're all done at atmospheric pressure. There's a very broad peak we're looking at, Q branch, with a bunch of different peaks in it. But in our atmospheric or breath measurements, we're also going to have lots of other species present. In particular, ammonia, carbon dioxide, and methanol. So ammonia in green here, carbon dioxide in blue, and methanol in red, and black is our isoprene trace, also show up in this frequency range right near our peak of isoprene. And so this is using some concentrations that would be similar to what we'd anticipate for a breath measurement, based on previous measurements of human breath at an atmospheric pressure. And we can see these simulations show that the isoprene peak is going to be swamped by the wing of this ammonia peak. Ammonia is present at somewhat higher concentrations and has a stronger absorbance peak than the isoprene. And so if we do this at atmospheric pressure, we're going to have a hard time distinguishing our isoprene peak from the ammonia peak. So the solution to this is we need to measure our isoprene at lower pressure, which decreases the pressure broadening of the peaks and then allows us to much more easily distinguish the isoprene from the ammonia and carbon dioxide and methanol that are present. And so we need to go back and recheck our measurements at low pressure. And so my students and I did this. We still get very linear behavior, which is excellent. And we've repeated the Allan deviation measurements. The Allan deviation, we need to average for a little bit longer to get down to our lowest values. And also the lowest value is about 3 times higher than it was at atmospheric pressure. And the reason for this is just that the signal is weaker at lower pressure because of the decreased pressure broadening. The Q branch actually has less overlap, and so the peak actually gets smaller. But still within the range where we can observe breath measurements. And so that was the next thing that we did. We collected breath samples from a volunteer after a brief exercise. The exercise increases the concentration of isoprene. We kind of want to give ourselves the best chance to actually observe the isoprene. And collected samples in a Tedlar bag like this. And then the sample was moved from the Tedlar bag into our multipass cell. And we did this at the lower pressure of 190 Torr to prevent the spectral interference and no further processing of the sample. And then we took an average of 10 spectra of the sample and then 10 spectra of a background of nitrogen at the same pressure. And so here are our spectra that we collected. So these are 2 different samples that we collected. The black trace in each of these spectra is our experimental measurements. The red dashed line is our best fit using least squares fitting of here are carbon dioxide, ammonia, and isoprene peaks. So on this spectrum, we only see the very edge of the carbon dioxide peak, pretty clear ammonia peak, and a pretty clear isoprene peak. And so from the residuals of our fits are very similar to what we got from fitting our standard isoprene sample. And so that's encouraging as well. These are very low concentrations, so you can see these aren't the greatest fits you've ever seen, but we can retrieve concentrations So in sample A, here are the results. Isoprene, in particular, is what we care about, is present at about 130 parts per billion in the first one, and in the second one at about 210 parts per billion. And so this shows that we can actually take a real sample, don't even have to do any extra processing to it, and we can retrieve isoprene concentrations. Now, we weren't able to compare this to a different method. We didn't have another method we could use readily available to double check, like mass spectrometry or something like that. But these concentrations are reasonable. They're within the normal range of isoprene concentrations that have previously been measured in humans. So, what can we do to improve this? Well, one thing we can do is use a couple of stronger bands of isoprene at about 900 wave numbers. They're about 2 times stronger than the one that we've chosen. It just doesn't work with the laser that we currently have. We could also use a pre-concentration system, which would drastically improve signal, but would also increase complexity and decrease our temporal resolution. Or we could use wavelength modulation spectroscopy. And we have attempted this with our current setup, but it doesn't work with the particular laser that we have. It's not able to modulate sufficiently deep for the width of the peak that we have, since it's a pressure broadened Q branch. So in conclusion, isoprene is an important molecular monitor in atmosphere and human breath. We have built a QCL-based spectrometer capable of measuring parts per billion level concentrations of isoprene, and demonstrated that we can actually measure this in a real-world sample of human breath. And we would like to further improve this, and so we could actually use this for atmospheric measurements, which would be excellent to do. So real briefly, I'm a little over time, but I'd like to acknowledge my students who worked on this, Jake, Melanie, and Grace. Yeon also worked a little bit on this, but she's not an author on this one. And also thank Connecticut College for funding, and then encourage you to check out our paper in Applied Physics B about this work. Thank you very much.